friends, good afternoon and welcome to EduSat Live Lectures. Dear friends, today in Immunology we will be talking about complement systems. To discuss this topic we have with us our subject expert Dr. Hardeep Kaur. Dr. Kaur is Assistant Professor in Department of Zoology in Ramjas College, University of Delhi. Without further ado, I would like to welcome ma'am to our studios and request her to start the lecture. Welcome ma'am.
Thank you, Amrit. Good afternoon, viewers. In today's lecture, we will be dealing with complement system. It's a very important system, a very important part of immunology. Now, before we hop on to complement system, let us recall what we have learned till now. So, we must understand what is the lymphatic system. We did it in our previous lectures. The lymphatic system is composed of lymph vessels, lymph nodes and organs. The function of the system include the absorption of excess fluid and its return to the bloodstream, absorption of fat and the immune system function. Lymph organs include the bone marrow, thymus, spleen and lymph nodes. Now, another important thing is immunity. Now, immunity is the body's capability to repel foreign substances and cells. The non-specific responses are the first line of defense. Highly specific responses happens to be the second line of defense. Now, we also remember that the physical barrier which protects us from most of the pathogen is our skin. That's the first line of defense for us. Now, after that, once a pathogen penetrates the skin or the epithelium lining of a respiratory tract or digestive tract or urinary tract, the pathogen, the body, there is inflammation that takes place in the body. The damaged cells hereby release histamine, which increases the capillary blood flow into the affected area, hence the area becomes heated up and reddened. Now, if we remember, there are 5 cardinal signals which show acute inflammation: rubor, that is redness, calor, increased heat, tumor, swelling, dolor, pain and functionalesa, that is loss of function of that particular organ. Now, what does these do? Generally, the heat that is produced during the inflammation, it inhibits the growth of pathogen in that region. It further promotes healing and raises the mobility of WBCs, that is white blood cells, and increases the metabolic rate of nearby cells. Finally, the monocytes come there, clean up the dead microbes and the cells and the debris. Now, the question arises that what happens if the inflammatory response is not able to contain the infection? What will happen if all this first line of defense, all inflammation, acute inflammation, everything that has happened is not able to contain the infection and the pathogen is still is very much alive inside your body? It is very much good in your body, good to go. Then what will happen? Then the answer to this question is the complement system. The complement system, along with the other machinery of the immune system, is here to protect us from this growing pathogen inside your body. So, let us come to what is a complement system in that case. Now, complement, which is generally given as a capital C, it refers to a system of factors which are occurring in normal serum. Now, what are these complement actually? They are proteins or glycoproteins that are produced in the liver. They are kind of a protective proteins. Now, what do? They directly kill microbes sometimes. They can supplement the inflammatory response, or they can work along with the immune response. So, they can work at 3 levels, either directly killing the microbe or supplementing the inflammatory response or working alongside the immune response. Now, the reason we call these proteins as complement system or these proteins as complement proteins is because they are actually complementing the action of the antibodies or the immune response. Immune response is functioning, the antibody is functioning, doing its work, but complement system is enhancing or complementing the effect of the antibody, hence called the complement system. Now, many a times any antigen antibody reaction, that is an immune complex that is formed inside the body and is moving around in the blood, triggers the complement system. At times it may be the microbe directly being affected by the complement, or at times the antigen antibody interaction, that is the immune complex, is triggering the complement system activation. Now, with this we come to the history of the complement system, who all had worked hard to make us understand what complement system is all about. Now, the first thing, Hans Buchner was the first to discover a substance in the blood serum that was capable of destroying bacteria. That means it was leading to bacteriolysis. This he came up in 1889. He called this substance as alexin, and he found that this substance is highly heat unstable. Now, if you subject the serum to heating at 55 degrees Celsius for 1 hour, this serum will lose its this bacteriolytic activity. That means there was something in the serum which gets broken down if we heat the serum. So, this heat labile thing was named as alexin. This name was later changed to complement by Paul Ulrich in 1854, who worked from 1854 to 1915. Now, post this, R.F.J. Pfeiffer in 1894 found that the live cholera bacteria could be injected into guinea pigs who have been previously immunized against the cholera. So, these guinea pigs have already been immunized against this pathogen, cholera pathogen, and now the blood plasma from these animals, once it was added to the live cholera bacteria, it was found the bacteria underwent lysis. That means bacteriolysis took place. This bacteriolysis could be easily inhibited by previously heating the blood plasma. This phenomena was later called as Pfeiffer phenomena or Isai-Pfeiffer phenomena. Now, later J. Baudet in 1895 found that the bacteriolytic effect of the specific antibody is significantly enhanced in vivo by the presence of certain innate serum components, which were later known as complement. Now, this is the history of complement. So, Baudet actually got a Nobel Prize because of his discovery of this component. Now, complement hereby kills microbes in three basic ways, three different ways. It either opsonizes the particulate antigen, it can opsonize the wear out cells also, it can opsonize certain erythrocytes also, who have, like we all know in our previous lectures, we have discussed that erythrocytes have a lifespan of 120 days. So, after 120 days they need to be removed. Now, for the removal of erythrocytes, the complement proteins can opsonize. Opsonization although we had done it in our previous lecture, it is like something that coats something to make it more visible. For example, here the complement protein is opsonizing or coating the microbe in such a way to make it more visible for the immune system to kill this pathogen. Now, this is one of the very important function of the complement proteins. Now, the second function is the inflammation by binding to specific receptors on the cells of immune system. Now, once we are dealing with the various pathways of complement activation, we will realize that there are certain complement fragments or complement protein fragments that actually go and bind to certain cells, which initiates an inflammatory response in our body. Now, this will become more clear as we go into the activation or the pathways of complement activation. Now, the third important function of complement is the lysis of cells. These could be worn out cells, this could be the erythrocytes who have lived their life, or they could be bacteria, viruses, pathogen, anything. The lysis of cells is initiated by the complement system. Finally, the immune clearance of immune complexes. Now, once the antigen and antibody complex has formed, now it cannot go on living forever inside our blood. It has to be removed. This immune clearance of immune complexes from their circulation is done by the complement system. What do? They clear these immune complexes from the blood and deposit them either in the spleen or in the liver, and hence immune clearance takes place. We cannot go on having immune complexes in our blood forever. Once the work is done, it needs to be removed. Now, these complement proteins also work as a triggered enzyme cascade system. Now, what is a triggered enzyme cascade system? Cascade, if you understand, is when one reaction triggers another reaction which triggers another one and so on. So, it is like one reaction triggering another, this once gets activated triggers another, this gets activated triggers another. So, this is called a cascade mechanism. Now, generally such systems grows exponentially, they have a very fast growth. Now, few of the examples where you might have gone through the cascade mechanism is, for example, in the coagulation, the fibrinolytic system, the kinase system. Similar system also takes place in the complement. Now, the main components, if we understand, if we want to know the components of the complement system, are around 11 proteins which have been designated as C1 through C9, along with certain factors called factor B and factor D. All these proteins are present normally among the plasma proteins in the blood, as well as among the proteins that leak out of the capillaries into the tissue spaces. Now, most of these circulate as precursors or proenzymes. Now, what do we understand by proenzyme or precursor or a zymogen? It means that generally in this form, these proteins are in an inactive state. Now, to activate these proteins, it needs to, there is some inhibitory molecule or on these proteins that needs to be removed. So, technically the protein lysis or proteolysis of a part of the inhibitory fragment of these proenzymes later leads to exposure of the active sites of these enzymes. So, technically a proenzyme can be made an active enzyme by removal of an inhibitory fragment. This is very important because all these complement proteins by and large are always present as a proenzyme, and for their activation a inhibitory fragment from their protein fragment or from the protein structure has to be removed to make them an active enzyme. Now, sera from different species have complement components in different proportions. In some, certain protein would be more, in certain cases the other protein would be more, but are functionally effective even when you exchange the complement proteins between various species, from one species to another. Even if we exchange these complement proteins, they are equally effective. Now, coming exclusively to the complement components, the complement proteins are often designated by an uppercase letter C, the capital C, and this capital C is inactive unless it splits into its products. So, if I say C1, it means the first complement component we will designate as a capital C and we will write 1 next to it. Now, if it gets split and it has to be activated, the active products are designated with a lowercase a or b. For example, if C1 was the inactive or proenzyme C1 protein, then its active form is C1a and C1b. Now, generally it has been found that the smaller fragment post cleavage is designated as A and the larger as B. However, the C2, that is the second component of the complement system, is an exception. Here C2A is larger and C2B is smaller in this case. Now, the larger fragment binds to the target and the smaller fragment generally diffuses out from the site, and it leads to initiation of a localized inflammatory response. What does it do? This smaller fragment goes off into the bloodstream, gets bound to the receptors of certain cells, and these cells are the ones which are responsible for inflammatory response. So, these smaller fragments ooze out or goes out from that site and then finally binds to those cells which are going to lead to an inflammatory response. So, the larger fragment has a key role to play. It is the one which is causing the entire initiation of the complement system. The complement fragments interact with one another to form functional complexes. So, technically if one protein of the complement system gets activated, it will complex to another protein, activate it. Now, this complex will be acting as an active complex, it will activate the third protein. the third protein on getting active after bound to this complex will become another active complex. So, technically all these components bind to each other and form complexes or the functional complexes. Complexes that have enzymatic activity are designated by a bar over the number or symbol. For example, if I say C4B2A with the bar, it means the active component of C4, which is C4B, and the active component of C2, that is C2A, have formed a functional active complex, and this complex together, both these proteins are joined together and they are in an active state. They are enzymatically active. So, if we have to show it, we will show it as C4B2A with a bar over it. The bar shows that this complex is active in nature, or for example C3BBB. So, it is 3B small and B capital and a small b, means it is the active components of the C3 protein and the active component of the B factor together forming complex and are in a active state. So, this is how we designate complement proteins. Now, after this is clear, we will come to the complement activation. The complement pathway can be activated by either of the two different pathways. The first pathway is called the classical pathway, which is a specific immune system response which involves antibodies. Now, the other pathway is called the alternative pathway, which is not specific. It belongs to the innate immune system. There is another pathway which has come up, which is called the lectin pathway. Now, by all these three pathways, the complement activation takes place. Now, each of these pathways have a specific role. As I said, the classical pathway is more to do with the antigen antibody response. For example, if there is an immune complex that has formed, then mostly the classical pathway will get activated. So, it forms a part of the humoral immunity. However, the alternative pathway has more to do with the innate response. It is not specific. It sees a pathogen coming inside and the alternative pathway starts. It does not require the presence of immune complex. Another pathway which I said, which is a lectin pathway, although too much details of lectin pathway is not known, but it is a predominant pathway specifically when we are talking about those microorganisms which have mannose residues on the membrane. So, by means of all these three pathways, the complement activation takes place. Now, if we come to the classical pathway, the classical complement pathway can be activated by formation of soluble antigen antibody complex, which we call it as an immune complex. Now, binding of an antibody to an antigen on a suitable target like a bacterial cell initiates this pathway. Now, as I told you that this pathway gets activated only in the presence of an antibody, that is only in the presence of an immune complex. An immune complex is an antigen and antibody. Now, what are the antibodies which are responsible or have a role to play? The antibody which is responsible is the IgM and the IgG. Now, IgG also have subclasses. So, IgG1, IgG2, IgG3 are the only antibodies which are involved in the classical pathway. Now, the components which are involved in the classical pathway are the C1 component, the C2 component, the C3 component and the C4 component. Now, the C1 exists in the blood serum as a molecular complex. Now, it contains three important parts. There is one molecule of C1q, two molecules of C1r and two molecules of C1s. So, technically, one molecule C1q, two of C1r, two of C1s, together 5 of these proteins form C1. Now, it is most of the time moving around in the blood serum. Now, whenever there is an immune complex formation, the antibody, the constant region of the antibody has a binding site for C1q, which is a part of C1 complement molecule. The C1q molecule itself is composed of 18 polypeptide chains that associate to form 6 collagen-like triple helical arms. the tips of which bind to exposed C1q binding site. If you remember, there was a CH2 domain in the constant region of the heavy chain of the antibodies. So, the second domain in the heavy chain constant region of antibody is the site where actually C1q binds. Now, the binding of C1q further activates C1s and C1r. Now, once C1 gets activated, it activates two other complement proteins, that is protein C4 and protein C2, by cutting them into two fragments each. As I have already told you, when C4 needs to get activated, it will get activated in the form of by removal of an inhibitory molecule. So, C4a is the smaller fragment of C4 which is removed, and C4b is the larger fragment which has a role to play in the classical pathway. Now, once C4 is cleaved into C4a and C4b, this cleavage takes place by the action of C1s. So, you understand the C1q binds to the CH2 domain of the antibody, the C1s and C1r are joined to it. The binding of C1q activates C1s, C1s further C1q activates C1r, C1r activates C1s, C1s further activates C4. Now, activation of C4 involves removal of C4a. The active of C4b now binds to the microbial surface. It attaches to the target surface next to C1, C2 proenzyme, now next to C1. Now, once it has attached to next to C1, another proenzyme, that is C2, comes and binds to that region where C4b is attached to the microbial surface. Now, once C2 comes and binds at that region, this C1s again cleaves this inactive C2 into C2a and C2b. So, if you remember, I told you C2 was an exception where C2a was the larger fragment and C2b was the smaller fragment. The role of C2b is not very well known. So, C2b goes off, the C2a now gets bound to the C4b which is already present on the microbial surface. Now, both C4b and C2a bind together on the surface of the bacteria to form a complex which is called C4b2a. As you can see in this slide, we are having 4b and 2a. Now, if I show it with the bar, that means this is an active complex. This complex is also called C3 convertase because it has an enzymatic role to play to activate the next protein in the pathway, that is the C3 protein. The smaller fragments C4a and C2b, they just diffuse away. Now, C4a actually is an anaphylotoxin, that means it initiates an inflammatory response, the details of which will come later in our lecture. Now, further, as I just told you, the C4b2a, the active C4b2a complex is a C3 convertase. Its function is to activate the C3 protein. This activation is done by cleaving C3 into C3a and C3b. Now, it is very important to note that a single C3 convertase generates nearly 200 molecules of C3b. Not all C3b would be utilized here, some would be used as an opsonin later. The details again I will come to it in my later half of the lecture. Now, what happens? Now, some of the C3b will attach to the C4b2a complex to form a tri-molecular complex. As you can see, the complex is C4b, 2a, 3b. Now, as I am showing a bar over it, it means it is an active complex and is also called C5 convertase. Now, C5 convertase, as the name suggests, will convert C5 to diffusible C5a and C5b. Now, C5b will get attached to the microbial membrane a bit closer to this already formed trimolecular complex. Once it gets attached to the microbial surface, it will now then bind to C6, C7, C8, C9 and will hence initiate a membrane attack complex. Now, as you can see in this slide, the C5b which is now getting bound to the microbial membrane, it is now getting attached to C6, C7. Now, this C5b67 is now getting attached to C8. Now, if you see C5b678 is forming a complex. This C5b678 will react, or rather will combine with multiple C9 proteins, and this C9 along with C5b678 will form a membrane attack complex, which is very, very important for killing the pathogen. Now, C3, as I just told you, is the most abundant protein of the complement system. About 1.3 milligrams of C3 is present per ml of the blood. Some of the C3b does not bind C4b2A, instead it acts as an opsonin. As I told you, opsonin is something which coats the pathogen. So, C3b, which is a residual C3b, will now coat the pathogen and will lead to killing of the pathogen. So, as an opsonin, C3b molecule will bind to bacteria and will increase its phagocytosis by about 1000-fold. With this, I will come to the end of the first part of my lecture. I will continue this lecture and the other pathways of complement activation in my next lecture. Thank you very much. Welcome back, viewers. Now we will be discussing the rest of the complement activation pathways, how complement deficiency leads to problems in our life. So, let us come back, revisit the earlier lecture where we were discussing the classical pathway of complement activation. If you remember, as you can see in this slide, this is the complement pathways, the glance towards the complement pathway. Now, in the upper half you will see the classical pathways there, wherein we can see that this how antigen antibody complex which is formed in the blood is activating the C1 complement component, which further in fact activates C4 and C2, which forms a C3 convertase. C3 convertase further acts on the C3 complement protein, which joins together to form a trimolecular complex which is called a C5 convertase. C5 convertase, as the name suggests, has a role to play to act on C5. The C5 changes to C5b, joins to C6, C7, C8, C9 to form MAC, that is membrane activation complex. Now, important thing is this C3b protein which is formed and the rest of the C4a molecule, C3a molecules, which we have not discussed in our previous lecture. So, if we come to it, this C3, as I was telling you, is the most abundant protein of the complement system, which is about 1.3 mg per ml of blood. Now, as I was just telling you that not all of the C3b is going to bind to the C4b2a, the active C4b2a complex, that is the C3 convertase complex. So, some of it actually remains as an opsonin because there is so much of C3b present already in the blood. some of it does bind and continue in the classical pathway. The rest of it act as an opsonin. It forms a coating on the bacterial surface and promotes the bacterial lysis by acting as an opsonin. Now, coming to the C3A, C4A and C5A proteins, which we haven't spoken about, as I was telling you, the C3A or C4A or C5A are those small components which were removed to activate C3, C4 or C5 complement proteins. Now, what do? They increases the inflammatory response by binding to the mast cells. Now, here you can see there is a mast cell is having histamine granules. Now, what does it do? The C3A or C5A or C4A, they will go and bind to the receptors on the mast cells. Mast cells on getting activated or getting triggered by this complement protein leads to release of this histamine. Now, histamine we had already known how histamine affects it. So, technically these C3A, C4A and C5A, they may not be directly acting in the complement cascade, but they have a very important role to play. They are called anaphylatoxins because they are initiating the inflammatory response in the body. Now, with this we come to the alternative pathway. As I told you in my previous lecture that the complement activation takes place by classical pathway, by alternative pathway or by the lectin pathway. Now, the alternative pathway does not require the presence of the immune complex. It is part of the non-specific defense because it does not need any antibodies to initiate the pathway. So, hence we say that the alternative pathway is belonging to the innate system. Now, the crux of this thing is that all these pathways are finally generating C5b molecule, which finally leads to formation of the membrane attack complex. Now, this also, of course, the alternative pathway is also leading to the generation of C5b. It is a bit slower than the classical pathway and it involves 4 major serum proteins, that is C3, factor B, factor D and properdin. Now, if you can see in this slide, we will go through this entire pathway one by one. As you can see, the C3 is getting activated, it is joining to factor B, then it is joining to factor D, then properdin has a role to play which leads to formation of C5 converters, and finally it is the C5 converters is causing the activation of C5 and the active C5b is leading to the membrane attack complex. Let us go one by one to all these steps. So, the first step is the C3. Important is that C3 needs to get activated. We had already said that C3 it spontaneously undergo dissociation many a time. So, we have surplus C3b all the time in the blood. Not only that, it has unstable thioester bonds which is causing this C3b to spontaneous slow hydrolysis to C3b and C3a. Now, C3b is able to bind to the foreign surface antigen. The foreign surface antigen could be a bacteria, could be a virus or could be a worn out cell also. Now, the mammalian cells, important part is that is it just joining to the pathogen? Is it not joining to your own body cell? Yes, it is joining not only to the pathogenic cell but also to your body cell. But thing is that mammalian cells contain sialic acid which inactivates C3b, which is unlike the foreign cells. Foreign cells are devoid of sialic acid. So, because sialic acid is less in the foreign cells, the C3b actively binds to the foreign cells. However, to your own body cells, because you have surplus of sialic acid on the membrane, it does not get bound. It just gets dissociated from your own body cells. So, we have understood that all this surplus C3b is actually getting bound to the pathogenic membrane, pathogen's membrane, because of the less or absence of sialic acid in most of this pathogen's membrane system. Now, once C3b binds onto the surface of a foreign cell, another plasma protein called factor B comes and joins this C3b molecule. Now, after joining, as I just told you, the binding of C3b to factor B allows another protein enzyme which is called a factor D enzyme, which what does it does? this factor D cleaves factor B to BA and BB. Now, factor BB will remain bound to C3b molecule. So, it forms a complex which is called C3b BB, and we put a bar over it to show it that it is an active complex. So, this active complex, that is C3b BB with the bar, is called C3 convertase because it has an activity. Now, it will be acting on C3 again. So, the factor, and so while BA and the factor D is dispersing away. So, technically the binding of C3b to factor B, it is allowing a protein enzyme factor D to cleave factor B to BA and BB, and this factor D, after its work is done, that is after BB is formed and it has joined to C3b, this factor D will go off, the factor BA will also go off. Now, C3 convertase, which we call it as C3b BB with the bar, has a half life of only few minutes. However, binding of properdin stabilizes it and increases its half life to almost 30 minutes. So, technically the function of properdin is to stabilize this C3b BB with the bar, that is the C3 convertase, the active C3 convertase enzyme. Now, the C3 activation complex or the C3 convertase, which we call it, it causes production of more and more C3b. Now, when more and more C3b is formed, some of it will get bound to this active complex, the C3 convertase complex or the C3 activation complex, as some people say. So, now we call it as C3b BB 3b with the bar. Now, this trimolecular complex is a typical C5 convertase. It's as the name suggests, it will act on the C5 complement protein. Now, this allows the initial steps of this pathway to be repeated and amplified, this particular step. Now, almost 2 × 10⁶ molecules of C3b can be generated in just 5 minutes. So, you can understand so many, this pathway, this step is so quick, and this pathway is leading to so many humongous amount of C3b molecules, and we know C3b has a key role to play in complement, not only in the pathways but also as an opsonin and other roles also. Now, when an additional C3b binds to the C3 activation complex or to the C3 convertase molecule, it converts it into a C5 activation complex. That means it is now called C3b BB 3b, and we put a bar over it. So, it is a tri-protein complex or tri-polypeptide complex which has a role to play, which will act on the C5 molecule. Now, this C5 activation complex, or we commonly call it as C5 convertase, will cleave C5 into C5a and C5b. Now, we all know all these pathways are finally leading to production of C5b. Now, C5b will then bind to the antigenic surface and will hence begin the production of the MAC complex. We know we had done it before also. The C5b will now react with C6, will react with C7, C8, C9 and a column like structure or a pore like structure, which will deal later, which we call it as MAC or membrane attack complex, will finally form. So, what we had done is, by both these pathways we are coming back to the region where we are getting C5b. Both pathways are converging to the active C5b molecule, which is binding to the pathogenic surface. There is another pathway which we call it as a lectin pathway. Now, this lectin pathway is not as maybe not too much is known about this lectin pathway, but still it is a very important pathway. Now, lectins are those proteins which bind to the carbohydrates. They do not, again, why this lectin pathway is important? This lectin pathway is somewhere in between both the classical pathway as well as the alternate pathway. It does not require the antigen antibody complex, which is very unlike the classical pathway, but once it gets activated, it almost follows the same steps of the classical pathway. So, few things it has taken up from the alternative pathway and few things it has taken up from the classical pathway. So, it does not require antibody for activation, Now, many bacteria have many mannose residues on their surface. These are the carbohydrate molecules present on the surface. Mannose is a carbohydrate. Now, these are the carbohydrate molecules present on the surface of the bacteria. Now, lectin based complement system begins with a mannose binding lectin, which we call it as MBL. Now, what happens is the MBL or mannose binding lectin is present in the serum. This MBL will go and bind to the mannose residues present on the pathogen. Now, MBL is an acute phase protein which is generally produced in an inflammatory response. Now, after binding to the antigenic surface, the MBL will react with MBL associated serine protease, which we call it as MASP or MASP. So, after binding of MBL to the MASP, now MASP functions in effects like activated C1q-R2S2. Now, what was C1q-R2S2? That was a C3 convertase. Now, this was very important and this is leading to further steps. Now, the rest of the steps of this pathway are almost exactly like the classical pathway. So, if we give a glance to all the three pathways, we will see the classical pathway which is getting activated, as you can see in the slide, the classical pathway which is getting activated by the antigen antibody molecule is finally leading to the production of active C5b molecule. Similarly, the alternative pathway which gets activated due to the microbial surfaces is again forming a C3b-Bb3b complex which is called C5 convertase, which is again activating C5, forming an active C5b molecule. This active C5b molecule is joining to C6, C7, C8, C9 to form the active MAC complex. Now, on the other hand, the lectin pathway has few of the characteristics of the alternative pathway, wherein it is taking up the microbial activation is by the microbial cell wall. The MBL present in the serum is joining to MASP, which acts like a C1 like complex. It is just acting like C1q-R2S2, which is almost analogous to the first component of the classical pathway. It now acts on the C4 components and activates it, and the C4b, the active C4b is formed, which along with C2b will form a C4b, which along with C2a will form C4b2a, that is C3 convertase, which will again convert C3 molecule to form C5 convertase, that is C4b2a3b, which will act on C5. The active C5b will react with C6, C7, C8, C9, which will finally form the MAC complex and will kill the pathogen. So, this is the entire crux of all the three pathways. All the three pathways are converging to form an active C5b molecule, which finally forms MAC, that is membrane attack complex. Now, what is this membrane attack complex? The MAC is responsible for the lysis of the cell. It involves, as I told you, C5b, C6, C7 and C8 and C9 that forms a macromolecular complex. This complex replaces membrane phospholipid, forming a large transmembrane channel. Through this channel, cytoplasm can rush out and water can rush inside the cell. Now, you understand, if the cytoplasm rush out and water enters it, the osmotic balance of that cell is just not there. It is compromised. It has been compromised. Now, because the integrity of the cell has compromised, the cell will eventually die. Now, these are some of the biological effects of the complement protein. Now, interaction of the complement proteins and protein fragments also takes place with various receptors on various cells of the immune system that control both innate as well as acquired immune responses. As you can see, if we are talking about cell lysis, the most important complement products which are responsible for cell lysis is the ones which are forming the MAC complex, that is C5, B, 6, 7, 8 and 9. Now, there are certain, like in the inflammatory response if we are talking about, we have degranulation of mast cells and basophils. It is due to certain anaphylotoxin molecules. These are C3A, C4A and C5A. we had already discussed that how these molecules go and bind to the receptors present on the mast cells and basophils, how degranulation of mast cells and basophils takes place, how histamine is released, how it increases the vascular permeability, and so on and so forth. So, inflammation takes place. Now, degranulation of eosinophils is also due to C3a, C5a. The extravasation and chemotaxis of leukocytes to an inflammatory site is the responsibility of C3a, C5a, C5b, 6, 7. Aggregation of platelets takes place due to C3a, C5a. Now, release of hydrolytic enzymes from neutrophils takes place by C5a. Release of neutrophils from bone marrow, C3c. Inhibition of monocyte or macrophage migration due to BB. Now, important thing, opsonization of particulate antigen. We know that we had discussed how C3b is very important. C4b, IC3b are also certain components which are also involved in opsonization of particulate antigens. Viral neutralization. Again, role played is by C3b, C5b to C9, C6, C7, C8, C9, that is a MAC complex. The solubilization and clearance of immune complexes, or also called immune clearance, is solely done by C3b molecules. So, we see that every molecule of the complement system has a role, a specific role to play, and this specific role is very much given to them. For example, if it is degranulation of mast cell, it has to be exclusively done by C3a, C4a, C5a. Now, if we are talking about viral neutralization or maybe the cell lysis, it is totally because of the MAC complex, that is C5b, C6, C7, C8, C9. So, technically every component of the complement system has a particular role to play. Now, how does this complement cascade get regulated? Now, important part is we cannot have this active complement proteins forever running in our blood. The nature of this enzyme is very labile, they are labile in nature. Now, that is the first reason how it prevents the prolonged complement activation. Now, also our serum has certain natural inhibitors of the complement, thus it provides a homeostatic regulation. A heat labile alpha neuroamino glycoprotein inhibits C1 esterase. Now, CAF, which is called conglutinogen activating factor, it provides a homeostatic control of C3 activation. So, if CAF is present, C3 will not go unactivated forever. It provides a regulation to the activation of the C3 component. Similarly, anaphylotoxin inactivator inhibits C3a, C5a. If it is not present, then what will happen? The body will always be in an inflammatory mode. There would always be an inflammatory response in our body. So, we do require an anaphylotoxin inhibitor or inactivator also. Similarly, beta 1H is a beta globulin regulator which regulates the alternative pathway amplification. So, again we cannot have uncontrolled amplification of the alternative pathway. This is managed by beta 1H molecule. Now, if this complement system is so important, then there must be certain diseases or problems if we do not have this complement system. So, if there is a genetic error or because of any what reason, some complement components are missing, it leads to a disease state in our body. Now, the deficiencies of complement system have been grouped in 5 groups. They are named as 1, 2, 3, 4 and 5. The group 1 has a deficiency of C1 inhibitor. So, understand, if C1 inhibitor is not present, C1 will go on getting activated and more of active C1 will be produced. So, technically the activation of complement system will be going unregulated in the absence of the C1 inhibitor. This condition is called hereditary angioneurotic edema, and generally it leads to edema of the mucosal membrane, mostly of the respiratory tract. Now, if the edema of the respiratory tract mucosal membrane, especially the lungs and the trachea takes place, it can be very fatal also. Now, the second group involves the early components of the classical pathway C1, C2 and C4. Generally, whenever there is a deficiency of these components, it would lead to a state which is called systemic lupus erythematosus, commonly called lupus or SLE. It could also lead to glomerulonephritis or vasculitis. There is another group of deficiency diseases, the third, which is called C3 and its regulatory proteins. Now, because of the deficiency of the C3 and its regulatory proteins, it leads to severe recurrent pyogenic infections. Now, the next group, that is the group 4, is the deficiency of component C5 to C8. Now, you understand component C5 to C8 are extremely important for MAC formation, that is membrane attack complex formation. Any deficiency of C5 to C8 will not lead to membrane attack complex formation. These membrane attack complex are kind of pores which are leading to final fatality of the pathogen. If these components are not there, we will not be able to clear or kill the pathogen. So, it leads to bacteremia, generally with the gram negative bacteria, diplococci or toxoplasmosis. Now, there is another group which is because of the deficiency of the C9 component. However, no particular disease has been found in this case. Now, with this I would like to come to the end of my lecture. Thank you very much.
On that note, I would like to thank ma'am for this very enriching discussion, and I hope, dear friends, that you have come to understand what complement system of immunology is all about. On that note, thank you, dear friends, for watching our show. Stay tuned and keep watching. Thank you.